am extremely pleased and honored to be able to introduce two of my colleagues who are giving our second keynote at today's conference. And their title is Restoring the Red Cedar, Tracking Water Quality Trends with the Red Cedar River Basin Monitoring Group. And so I'm going to introduce my colleagues Keith Gilland, who is an associate professor of biology here at the University of Wisconsin-Stout. His research focuses on the rehabilitation of both aquatic and terrestrial ecosystems of the upper Midwest, including sport fish habitat, urban stream and stormwater management, and prairie and savanna restoration. He received his doctorate in environmental biology from Ohio University in 2012 and began working at Stout in 2016. His work on monitoring the Red Cedar Basin has been funded by a series of grants from the Freshwater Collaborative of Wisconsin, and he has been participating in citizen science monitoring of the watershed through the Watershed Action Volunteers program with his students since 2017. He also serves as land manager for the Chippewa Savannas chapter of the Prairie Enthusiasts and site steward for their Dobbs Landing project site located along the Red Cedar River near Colfax. And then Dr. Nicole Hayes is an assistant professor of biology at the University of Wisconsin-Stout as well. Her research interests include water quality, under ice primary production, and cyanobacterial harmful algal blooms. For the previous 4 summers, she has worked with local undergraduates from UW-Stout, UW-Eau Claire, and UW River Falls to sample Menomin and Tainter lakes and study the cyanobacterial blooms that occur every summer. Alongside undergraduate researchers from UW-Stout, she has conducted under ice experiments to determine how much cyanobacteria growth is happening when the lake is frozen. Dr. Hayes graduated with her PhD in ecology, evolution, and environmental biology from Miami University in 2015. She continued studying water quality in green lakes in Saskatchewan and Minnesota prior to starting as a faculty member here at UW-Stout in 2020. So please join me in welcoming to the stage.
All right. Hello. All right, so thank you, Tina, for introducing us. Today I'm going to get started introducing you to the work that Keith Gilland and I have been doing for the last 4 summers as part of the Red Cedar River Basin monitoring group. But I want to take a minute and acknowledge that this is far from just the 2 of us that are working on this project. So our version of the Red Cedar River Basin monitoring group is funded by the Freshwater Collaborative of Wisconsin, and we work with an amazing group of other faculty members here at UW-Stout, Drs. Mandy Little, Julie Beston, and Julia Chapman. And we're also really fortunate to work alongside Professor Arthur Neeland and Dr. Tina Lee as part of the Lakes REU program. And so we're actually going to be talking about data from both of those while focusing on the Red Cedar River Basin. Just to orient you to what we're going to do today, I'm going to start by giving everybody a little refresher 101 on what a watershed is. Then Keith's going to take over and talk to you about what the Red Cedar River Basin Monitoring Group is, and then go through a series of student projects that we have supported to help train students to be the next generation of water professionals. And then I'll come back and wrap up and talk about some of the long-term trends, or shortish long-term trends that we're seeing with the data that the students have been collecting. All right, so first thing, I want to remind everyone of what a watershed is. So a watershed is simply the land area that contributes material to a given body of water. And so as a limnologist, I'm really interested in what's happening with water quality, but what's happening with water quality is reflective of a diverse group of activities that are happening on the watershed. So traditionally, we think about the impacts of things like deforestation and tilling prairies and conversion to agriculture as being a significant source of things like soil, nutrients to the water, when you can measure that as water quality. But we also know that human activities on the watershed can improve water quality. So transitioning from conventional tillage to conservation tillage, for example, can greatly improve water quality, reduce soil in the water, decrease nutrient concentrations. But other types of activities have implications as well. So if we're tile draining fields or tile draining suburban neighborhoods, we're greatly increasing the runoff of water-soluble nutrients like nitrate. When we look at our watershed, you can also see that there's urban environments, other ways that humans are impacting that water quality. Urban environments have very little little permeable surface. So when rain falls on those roofs and on those parking lots and roads, we end up with really flashy storm events that can really impact water quality. We also see a diverse group of different contaminants that run off from our urban environments. Kind of the classic one of that is salt runoff from our winters. But it isn't just human activity on the landscape. There are lots of natural processes that are also impacting water quality or mediating what's coming off the watershed and winding up in our lake. So two of the other things that we have been looking at as part of the Red Cedar River Basin monitoring group is how things like changes in wetlands and changes in streams can impact water quality. So for instance, we know that wetlands are important sites for trapping nutrients and soil on the landscape and are really critical for keeping our lakes clean and healthy. But the reality is that a lot of our wetlands are at risk from emerald ash borers. Black ash trees are a foundational species in our local wetlands, and many of those black ash are dead and or dying. As a result, we're seeing changes in hydrology, which are likely to have downstream effects on water quality that haven't been realized yet. But we also see positive impacts of management and water and ecosystem rehabilitation. So things like restoring trout streams in order to improve fishing can also do things like stabilize banks and reduce sediment loads. And so when we're talking about water quality today, I just want to highlight that we're trying to think really holistically about everything that's happening in the watershed and how that leads to changes in water quality, and specifically how we can train students to be very multidimensional, to be able to consider all of these different factors that affect water quality. And then I'm going to hand it off to Keith, who's going to talk about what's happening on the landscape.
All right, well, thank you. There we go. So I'll start off with just a word about Stout. And so Stout is what we call a primarily undergraduate institution, or a PUI. And it's a way people talk about different universities and their purposes and the groups they serve and what they're trying to do. And so while faculty at a school like Stout, we still have a research expectation in our jobs. I and others and other folks have different philosophies for how they approach this. I primarily view my role and my position in preparing students that come here for what's going to come next in their life, in their professional field. So while generating new knowledge in our field is an important thing to do, and it's an important thing you do as a scientist and as a faculty member, it's at times secondary to thinking about, like, well, how can these 18 to 23-year-olds get prepared for what's going to come next for them? And so since I've been here, my professional life has generally been finding myself in a place and finding what the degraded ecosystems are in that area and thinking about how can I get involved in repairing them. And prior to coming to Stout, I was in Southeast Ohio, and it's a coal mine area. So in my previous life, I was a coal mine reclamation and reforestation guy. And now that I've found myself in Menomonie, which maybe undersells, I'm delighted to find myself in Menomonie. When I think about, like, well, what are our degraded ecosystems around here? We were formerly a prairie and savanna landscape, and a lot of that has been lost. So I do a lot of work in that realm. But also, many of our surface waters are degraded. So how can we bring those back to regain some of that lost function? And so the Red Cedar Basin Monitoring Group, this is a group that I may butcher the history, but I will give the brief history of my involvement and what I know. So I will say back to at least 2012, this group has been in existence to monitor the natural-ish area out there. And my introduction to the Red Cedar Basin Monitoring Group was through Ted Ludwig, who a lot of us probably know. And Ted got me involved in this through my teaching of the Gen Ed environmental science class at Stout, with his shorts and his tall wool socks and his boots and his USPS hat and his boundless energy to find out about what's out there in the world. So we started with streams, but the Red Cedar Basin Monitoring Group has also done other monitoring. They've monitored bat echolocation studies in the past, eagle studies, so really trying to keep track of what's out there in the world. So this project is again based on asking questions about what is the state of natural stuff in our watershed. And so I'm not an agriculture guy, and I haven't been, and so I feel at times I'm sort of in a little niche area in the watershed where I'm interested in forests and prairies and streams, and that's fine, and I found that there's a place for that, and there's value in doing that. So this was one of the first years that Ted and I got out and did some stream monitoring. I think this was up somewhere along the Hay River at some part back in the spring of 2017. And so that leads us to where we are with this Freshwater Collaborative stuff and the Red Cedar Basin Monitoring Group and where it is now. So the Freshwater Collaborative of Wisconsin is a state funding program that started to get its first rumblings of coming into existence in 2018, 2019 with some groups from UW-Milwaukee that wanted to start something called the Freshwater University. And so the Freshwater University group had organizational meetings at the different, what we call the comprehensive campuses, now styled as designated as the Polytechnic campus in the state system, about what this could look like. And it was talking about getting resources, funding for resources. There was talk of buying boats and buying vans to tow them to go on field trips and making new classes and hiring new faculty. And so that eventually morphed into a first round of funding from the Wisconsin Economic Development Corporation. And we applied for the Red Cedar Basin Monitoring Group in that year, and that would have been in 2020, and we did not get that funded. And then it was COVID, and so it was really a double whammy of not great news. But we did get some funding to start doing some curriculum development and develop a new class. But then in 2021, we applied again for the first round of state funding for the Freshwater Collaborative, and we got about $72,000 for our first round of this project to fund. The grant was titled the Establishment and Support of the Red Cedar Basin Monitoring Group, and it covered summer stipends for 3 research students to get out and work with us and do projects of some fashion. And so this was our first year, and so we had 3 students. We had Brittany, Cal, and Dylan, and so our funding came in kind of late, as those of you who work with grant funding and a lot of governmental funding, a lot of times you find out at the last minute that congratulations, it's late April and you're going to get to hire 3 students to work for you this summer and figure out some projects for them to do. So we scrambled and we hired our students, and we sort of built on what we had already been doing with parts of the Red Cedar Basin Monitoring Group and with Ted Ludwig's work, and got out and did some snapshot monitoring across the watershed. And so all of our students were able to, in addition to the larger data sets we wanted to collect, everybody gets to take on their own project to try to build their skills in some way. And so I'm going to kind of go through these quickly because I got several years of these, and so some of these I'll talk a little more about. Some of these are more in Nicole's realm. And so in the first year, our student Cal Breeden, who is now out in Montana?
He might be back in Iowa now.
Might be back in Iowa. So Cal has since graduated and gone off into the natural resources working world. He took a stab at looking at some of these trout stream restoration projects that have been going on in Wilson Creek and Gilbert Creek, to look at some of the baseline data we always collect: dissolved oxygen, macroinvertebrate communities, all the general stuff you typically do with the Water Action Volunteers monitoring if you've done that, to sort of try to get an idea of some of these restoration projects are a little over 20 years old now, and so we're interested in, well, how well are they holding up? Because it's just one of those tenets of doing restoration is, how do you know your project worked if you don't go out and monitor it? And so Cal worked on that project, and so something you'll sort of see as we go along, that the good news is most of these trout stream restoration projects are holding up fairly well. So their macroinvertebrate communities are still pretty good. Their water quality, their clarity is still pretty good. So they're not in dire need at this point of a lot of maintenance. And so Cal presented his work at the Research in the Rotunda, where we go over to the state capitol and present their work to the legislators and other folks that are also gathered there. And this gets back to that idea of training for your future. And one of the big things that, there are some young people here, and young people, the youths of America have congregated with us. One of the important things to be able to do is not just to do the work, not just get out and be a scientist and collect your data, but to also talk about it to a larger group of people, why you're doing the things you're doing, what are you doing, and what did you find? And so that feeds into this idea that this is a training opportunity. And so Cal did his work. Brittany focused on some of the larger GIS data and land use cover and its impacts on water quality, and Dylan worked on the first round of some of the algal bloom stuff, and I won't talk about this. I'll leave that for Dr. Hayes to talk about when we get to her portion of the talk. So this was the first year, and it was kind of a seat of the pants. We're gonna do a science. What are we gonna do? Well, what are we already doing that we can plug students into? And it was overall a really great experience for the students. They've all gone on to be successful in their careers. They're in, the initial sort of 2-3 years out of college job fronts. Brittany is working for one of the county conservation offices over on the eastern side of the state. Cal is working out in Iowa for the state after working in Montana for a while for the state. So these have been really successful in building these students' career paths. So in 2022, we applied again and got funded for summer 2023. The title of this project was the Cross-Campus and Partner Expansion of the Red Cedar Basin Monitoring Project. And so in this round of funding, what we wanted to do is to try to bring in partners from other campuses. So one of the ways that the University of Wisconsin system is kind of set up amongst the comprehensives is that we all kind of have our own niches and our own lanes, and there's not a ton of overlap often in what different campuses do. So, here at Stout, we have a person that does wetlands, we have a water person, we have a terrestrial person that does plant stuff, we have an animal person. And so there are gaps that exist at all the universities in terms of what their folks do. So what we wanted to do here was to try to bring folks from other campuses who might not do a lot of wetland stuff or who might not do harmful algal bloom stuff on their campus to Stout where we do that stuff. And so we had 4 students that year. We had Ryder and Casey, and we had Cal back again, which having a little bit of overlap year to year is often great. And we had Delaney from UW Eau Claire. So we had our first cross-campus student. We had tried that year to hopefully hire someone from Oshkosh and someone from River Falls, but a lot of times, again, this funding comes in late and everybody got their internships already. So, we did what we could. And so Casey worked on an ecosystem metabolism study, so doing some ecosystem respiration and photosynthesis in the streams, and also taking the first stab at really trying to do something with the macroinvertebrate data. So getting out, collecting the bugs, getting them in alcohol, getting them back, counting, IDing down to the lowest taxonomic level that we could. And so, again, we sort of refined our stream set that we were working on here and really started focusing in on the restorations and also getting some data from sections of Gilbert that hadn't been restored yet. And so sort of reiterating what we've been seeing before, that these restoration projects, even though they're targeted at the trout fishery, really have lots of other benefits. They're good habitats, they're good bug habitats, they're stable, the water quality is clear, and so they're having lots of ancillary benefits to the watershed on top of what they were originally envisioned for. So I love urban streams as someone that kind of also has a landscaping background and a construction background. Like, I love the idea of urban streams. I love that we ask them to do a lot for us. We ask them to be part of our stormwater infrastructure. We ask them to be green spaces. We ask them to be habitat, and so we ask a ton out of them. And so I love some urban streams, and I love that we have Galloway here in town that has tons of possibilities. And so Cal started working on some more dedicated work along Galloway Creek, and especially where we have some corridor projects that we've partnered with the city and Menomonie High School and the Lower Chippewa Invasives Partnership to do some corridor work along these sites. And so Cal started working on sedimentation and water quality in Galloway Creek. And again, these were presented at the research in the rotunda. And I'll pick up the pace here so I can not eat too much of our time. And then we got funded for 2 more years in this round. And so this round for 2 years was for almost $400,000 total. And so the title of the grant's on the sheet, this continuation and expansion. And so in 2024, we hired 3 Stout students and 2 Eau Claire students. We had Ted and Anne. Anne gets her own special picture, I think, not just because she wasn't in this picture, because she was very, very good. So she deserves her own picture. And so we also brought in some more faculty. We brought in Mandy Little to work on this. And so the student projects in this round, we had some that focused on the vegetation communities in these trout stream restoration corridors, looking across the ages. And so what we're seeing is kind of in that vein of there's the part about the trout, but then there's the other parts. And so these are largely following some pretty cool, in that these are constructed environments, typical successional processes. At the first couple years, they're kind of weedy, and then they sort of transition into sort of a sedge-dominated, typical riparian corridor. And so Ted worked on this, and he presented this at the Wisconsin Wetlands Association, and he's working over near Madison for an ecological contractor now. Mike Buss really dug into this macroinvertebrate community data, so I won't bore you with, we finally got what's the name of the name escapes me, the graph? The ordination. Ordinations of how these communities fall out at different ages, and it turns out that there are some differences in communities across time, that there's different bug species in the different ages, and this is a really great project, and we're continuing to work on this and hopefully getting it out into publication. I won't steal the ecosystem metabolism and lake stuff poster that Nicole's students did. And then summer 25, we had another round of students last year. We had Grant from River Falls, we had Faith from Eau Claire, we had Elias and Kayana and Shelby from Stout. So we had a solid crew this year. And so they got out and worked on some of the black ash swamp hydrology stuff with Mandy Little to look at these changes in groundwater levels as the ash trees die off and potentially change their transpiration patterns. And Grant worked on sedimentation in Galloway Creek. This is a draft of our poster for the AWRA conference over in Milwaukee a little bit later. Students worked on zebra mussels. I'll flash through that. Zebra mussels, the microcystin data that Nicole is going to talk about. And then the other 2 parts of this grant have been scholarships to take part in this advanced freshwater experience class through Eau Claire. And so this gets back to the idea that every campus kind of has some slight differences. And so what we've been able to do is put on this 3-week course in the summer where the students spend about a week at Stout, at Eau Claire, and at River Falls and get introduced to all the different things they do there. So they go over to Eau Claire and they get some good groundwater experience. They come to Stout and they get some stream and wetland experience. They go over to River Falls, they work with the dam removal, the stream team over with the dam removal projects. And so you get exposed to things that you might not be able to if you were going to do your entire career at just Eau Claire or just Stout or just River Falls. And so it's, again, building those backgrounds of being able to say, like, yeah, I got a little bit of E. coli screening experience coming out of school. And like, yeah, I've done some stream hydrology. Like, yeah, I've delineated a wetland. I've got some background in all of that. And it's been an awesome experience for them. And then also as part of this grant program has been putting on a high school freshwater experience class. And so we draw from high schools around the area to come for a couple weeks, and it's a similar program at the different campuses. And so our students help lead that. So like you get a little bit of environmental education and wrangling folks to do things experience. And so these are all really geared towards just building our graduates' portfolios of experience to help them be successful in the future. And then the last, I'll leave off with, this is all kind of in service of my main love in life is managing and rehabilitating ecosystems and getting out and doing the work. And whether that's stream corridor projects we're working on along Galloway, whether it's this prairie and savanna stuff at Dobbs Landing or out at the Colfax Red Cedar Preserve, it's great to do that work, and I love doing it. I love chainsawing and burning and digging and planting. But it's also imperative that if you do that, that you do some sort of adaptive management program. And so that is also where this fits in. And so I have overshot my time by a few minutes, so I will hand off to my colleague to take us home.
Excellent. All right. So Keith just went through a high-level look at kind of all the amazing things that are happening in the watershed, and I'm going to transition a little bit and pull together some students' projects and some community data to talk about some, again, shortish, longer-term trends in water quality. So again, go back to that watershed diagram, and as Keith really highlighted, there's a lot of stuff happening in the watershed, and all of those different things are reflected in water quality. So we can measure, we can measure water quality, or we can measure soil and nutrients in the water. We can measure those urban pollutants in the water. So you would never run out of different factors to look at to assess water quality. As an ecologist, I tend to like to use biological indicators of water quality. And so today I'm going to be really focusing on kind of two areas of water quality. It's primary production, and then also a little bit about invasive species. So one of the reasons I like to talk about harmful algal blooms is because they are both an important part of ecosystem processes, but they're also part of how people interact with the water body. It's the part of water quality that we're most impacted by. So harmful algal blooms are composed of multiple different organisms. Often we classify them as algae and cyanobacteria. Most of the time in freshwater, we're thinking about cyanobacteria. We know that when we have a lot of impacts happening on the watershed, that we see increased nutrients in the water. And when there's increased nutrients, we have much higher rates of harmful algal blooms. We have a lot more plants growing in that water column. So that's important for multiple reasons. First, that's the base of our food web, right? So those organisms, those harmful algal blooms are feeding the things that are feeding the fish. But that's also often what people notice. So I teach an introductory environmental science class here at Stout. We talk about the lake a lot, and one of the things that comes up is it's green and it's stinky, right? And that is that harmful algal bloom. So that is often the first things that people are kind of exposed to when they're thinking about water quality. And so the kind of last bit of that is that some of the organisms that are involved in those harmful algal blooms are also problematic species that make toxins. And so that's what I'm going to spend most of my time talking about today. We're going to talk about how much harmful algal bloom is there actually in lakes Menomin and Tainter for the last 20 years. And then I'm going to drill in a little bit to a bunch of student projects where we've measured a single type of toxin in Lake Menomin for the last 3 years, talk about some of the patterns that we see there. And then I'm going to wrap up and talk about invasive species. So unfortunately, zebra mussels are now found in both Menomin and Tainter. So we'll talk about some of the student projects looking at distribution of those. And then I wanted to wrap up on a happier note and talk about how we do have invasive plant species in Menomin, but we also have high plant diversity. So I'm going to start here by talking about long-term trends in the amount of harmful algal blooms that we have. And so I've had students sampling the lakes for the last 4 years. We always go out and sample at the same points. So in Lake Menomin, we sample kind of near that deep point in the southern basin, right where the river leaves. And in Tainter, we sample kind of right in that southern basin. I always think it looks like a snowman, so at the base of the snowman, right where the basin narrows down again. We always sample at those spots. And so then what I did is I went into the Surface Water Integrated Management System, or SWIMS, which is the DNR repository of all data that has been collected at those same points. So much of the data from SWIMS at those 2 points in these 2 lakes is from volunteers, and so I'm really grateful to many of you for volunteering to collect water quality data, because I draw on that as well. And then last, I want to mention that I also pulled some data from the CLEAR Lab, or the Center for Limnological Research and Rehabilitation. And so I'm synthesizing data from a whole bunch of different groups. So to start at, I kind of want to orient you to how we measure and how we think about those harmful algal blooms. So what we actually measure in the lake is the amount of chlorophyll. So that's the pigment that is responsible for photosynthesis, but really simply you can measure the concentration in the lake, and it gives you an indication of basically how much stuff is there. And it can range pretty widely from lake to lake. So I gave you some pictures here to help orient you. So this is a picture of Lake Tahoe. I don't know if any of you have ever visited it before. It's sort of famous for limnologists because it is one of the clearest lakes in the United States. So very regularly, chlorophyll concentrations out in Lake Tahoe are between 1 and 2, right? So it is one of our clearest lakes. Up top here then, I have a picture of Lake Tainter, where you can obviously see that it is very thick and green. That is actually likely well in excess of 150 micrograms per liter of chlorophyll, but that's very high, right? So this is the sort of range that we're talking about. Oh, and I also wanted to mention that there are 2 students who have really led the chlorophyll projects. Again, we have Dylan up here in the top picture, and then Kayana down here in the bottom. They have run, between the 2 of them, hundreds of chlorophyll samples each. All right, and so now what I'm gonna show you is the data that was collected. So I'm presenting annual averages, although the annual average is really focused on the summer when we tend to see a lot more phytoplankton, so May through September, how much algae is happening. And I wanna orient you to 2 benchmarks as well. The lowest orange line is at 25 micrograms per liter, and that's the target level of chlorophyll that the EPA set when they kind of identified the Red Cedar River as being impaired as a result of nutrient pollution. I also have a value of 87 micrograms per liter noted because that's what the average chlorophyll was in Tainter, again, in the 90s when they identified this as being an impaired water body. All right, so now each of these points represents an annual average of the amount of plant material in the water, or the size of the harmful algal bloom, is how I think of it. When you can compare each of the lakes, Tainter versus Menomin, from 1995 up until 2025. And you can see that, obviously we haven't reached that 25 microgram per liter benchmark. I would say that would be a lovely benchmark. It maybe is a little aggressive for our potentially naturally eutrophic systems, but kind of the other positive takeaway is it doesn't seem like we're increasing in terms of chlorophyll concentration either. You do have to take this data with a few grains of salt because it's collected by many different people, sampling in different ways, but I did try to screen, so we had pretty similar methods over this whole time. But the other thing that you might know about Lake Tainter and Lake Menomin, if you've ever been boating on them, is that we often have a whole bunch of algae just sitting up at the surface of the lake, right? So limnologists were trained to sample lakes in a certain way, and that's go to the deep point and collect water from like 3 feet or 6 feet of depth. And that's what all of this data is, an average at like 3 feet or 6 feet of depth. But we know there's something else happening at the surface. So for the last few years, I've also had students collect surface samples, so basically scoop up what's sitting on the top of the lake. And what we find is that chlorophyll concentrations are much higher. There is a lot of algae just sitting on the surface of our lakes. if you've been on the lake, intuitively this makes sense. Unfortunately, you've probably witnessed that. These dark green or the light green dots represent that surface algae, or what's growing at 0 meters. And so you can see in every one of the years that we sampled, the amount of surface algae is higher than what's mixed into the water column. And in some years, like 2025, it is a lot higher than what we're measuring in the water column. And a lot of this is the result of the species composition that we have in both Menomin and Tainter. So we have a phytoplankton community that's dominated, or harmful algal blooms that are dominated by these two taxa. Up top we have Microcystis, down below we have a Phanazomenon. I like to call these two species out in particular because you can often identify them without a microscope. So if you have ever been out on your boat in the lake and you look down at the water and you see these tiny little green specks that look like tiny blades of grass, right, that your lawnmower throws off when you're done mowing, kind of sharp angles, that's likely in a Phanazomenon colony. And if you see little kind of green fluffy cotton balls, again, very small, but they're kind of green and fluffy with soft edges, that's likely Microcystis. These two taxa, they tend to float. They make little gas bubbles in their cells so they can sit up right at the surface and steal sunlight from everybody else. So our phytoplankton community composition is likely to float, and so kind of just looking at what's happening in the water column is maybe not reflective of what we're seeing in the lake. The other thing that I want to mention is that these taxa, so again, our Phanazomenon and our Microcystis, tend to be toxin producers. So unfortunately, both of them make a few different types of toxins, as does this organism up here, Dolichospermum, which we also see a fair amount of in Menomin and Tainter, but that one you can't see without a microscope, so I didn't mention it earlier. And so the other thing that I wanted to work on with students in my lab was looking at one particular type of toxin, microcystin. It's relatively easy to measure, and it's probably one of the more commonly produced toxins. And so we decided to start monitoring microcystins. Again, we went out to the deep site of the lake here. We're just going to look at Menomin, collected water at 6 feet of depth, and then sampled multiple times over the course of the summer. So again, this is that toxin, microcystin here, and then each of these points represents the concentration on one day that we went out and sampled. The colors represents year, the lighter blues are 22 and 23, and then that darker blue is this summer with 25. I want to note, again, I have this dashed orange line here. So this dashed orange line is at 8 micrograms per liter. Excuse me. At 8 micrograms per liter of microcystin concentration. So the EPA has done a bunch of studies looking at microcystin and the risk that it's posed. And in 2019, they put out these guidelines that suggested that above 8 micrograms per liter is when we should start being aware of these harmful algal blooms and kind of maybe start restricting our activities on the lake. So the EPA guidelines suggest that at 8 micrograms per liter, you should no longer swim in the water. Swimming, you are really likely to ingest a pretty significant amount of water, and at 8 micrograms per liter, they started to measure impacts on the liver and kidney of people that were ingesting that water. But they also said that if you are participating in a recreational activity where you're likely to ingest water, you should also think about restricting your activities. So things like tubing and skiing and jet skiing, where you might ingest water, probably not as much as when you're swimming. Maybe during the period of time when the toxins are above, or microcystin is above 8 micrograms per liter, you think about limiting your use. I do want to note that sometimes you hear folks talk about inhalation risk or skin contact risks associated with microcystin. As of right now, the EPA doesn't have any recommendations on that. So this is all just likelihood of ingesting while interacting with the water. So again, most of the time in early summer, the lake is well below that concentration we're concerned with. As we move later into the summer, concentrations increase, but in 2 of the years, they don't increase to the point that it's of concern. However, something happened in 2025 where we saw a pretty big increase in the amount of microcystin. Again, this was looking at what was happening at 2 meters. We also collected those surface scums at 0 meters, and likely this is what people are interacting with in the water column, right? Like you're swimming up near the surface, you're boating at the surface, and so these stars represent the microcystin concentrations at those surface. And so now we have 4 time periods that are spread out over the course of the month where the EPA suggests that there are potentially some risks associated with swimming in that water and potentially even recreating in that water. All right, so that was, we've been collecting this data every year, but also at the same time, I had a bunch of students that were really curious about how those surface scums or how those harmful algal blooms were moving all around the lake. So again, those of you who have been on the lake, you have likely seen this, right? Right up at the surface, you have what looks like green paint sitting on the surface. If you walk around the lake, you'll see that in some bays and along some edges of the lake, you have a whole bunch of biomass that's kind of shoved against the side, but out in the middle, it's clear, right? My samples were all from the middle of the lake. So starting 3 years ago, Ethan, Grace, and Ayers, and then this summer, Elias, were really curious about what is the distribution of those toxins across the surface of the lake? And so they came up with a sampling plan. They identified 26 points that are somewhat evenly spread across the lake. And what they did is they went out multiple times per year over the last 3 years and collected surface samples at each of those points. From there, we analyzed the microcystin concentration. And we basically used an approach to kind of average that concentration across space. So again, the center graph right here are those 26 places we actually have microcystin constants measured at. And then the color on this map represents kind of the average microcystin across the surface of the lake. When you're looking at this graph, color represents the concentration of that toxin. Anything less than that threshold, that EPA threshold of 8 micrograms per liter is sort of yellow, green, blue, and anything above that threshold is orange and red. I want to note this is on a log scale so that we do have a pretty rapid jump in concentrations once you transition to the red color. So we did this. We did this sampling in 23, 24, and 25, and so here are the 3 kind of closest together dates in those 3 years. And again, we sampled each of those times and have kind of averaged across the space of the lake. So my big takeaway from these mid-July samples are that at no point did we actually measure microcystin concentrations above that 8 micrograms per liter, right? So for the most part, all 26 of these sites were below 8. Some of them were certainly getting close, especially if you look in some of these in the eastern basins or down in the southern basins. Microcystins are increasing as our harmful algal blooms are starting to build up over the course of the summer. So we have spatial variability, but we also have variability between years, right? 2024 looks great. You could have been out swimming in the middle of that lake in July with very few concerns about this particular toxin. But we also wanted to look at what was happening in August. Because we know these harmful algal blooms build up over the course of the summer, we kind of wanted to track progress or bloom development over time. We only have data from this kind of late August sampling period in 24 and 25. And now what I assume is jumping out at you is that we have a lot of lake area that has toxins that are in excess of that 8 micrograms per liter. Again, we see a bunch of it down in the southern basin in both years and a little bit kind of over in these eastern basins. So kind of takeaway from this is that, you know, there are certainly parts of the year when, based on microcystin concentrations, there aren't any EPA guidelines that suggest that there's any risks associated with swimming or jet skiing, tubing, or skiing. But other parts of the year, we do have concentrations that are above that threshold. And so thinking about how time is going to affect risk associated with exposure to a liver and kidney toxin is probably something that needs to be considered as we're recreating in Lake Menomin. All right, and so now I'm going to kind of quick transition and wrap up by talking about the invasive species projects that I've had going on. So 2.5 years ago, there was the first report of zebra mussels in Lake Menomin. In that following summer, Jennifer Kahn started looking at zebra mussels, and then Kiana Erickson has continued looking at those zebra mussels. And basically what we wanted to do was just a survey of where zebra mussels are found in Lake Menomin. This first year, we took a very generous approach and we sampled habitats we were pretty sure we weren't going to find them in, just so that we had a pretty good estimate of zebra mussel population density across the whole lake. So each of these points represents a site of zebra mussel sampling, and then the color and size represents how many we actually found. So most of the lake is blue dots, right? This makes sense. Most of the lake has a mucky bottom. Zebra mussels don't like mucky bottoms. We would have been surprised if we found them there. But as you move closer to shore, where we have more hard substrate, which we know zebra mussels like, so this includes kind of rocks and riprap along the sides of the reservoir. It also includes boat landings and docks. So wherever there's hard structure, often human-made structure, we measured zebra mussels. I don't know if folks from Menomonie also remember that in the summer of 2024, the police department also pulled a truck out of the southern basin of Lake Menomin, and a truck is hard substrate, and we were trying to assess zebra mussels, so we went to the junkyard and we found this truck and we counted the amount of zebra mussels on it. And this truck had by far the highest concentrations of zebra mussels we found in the entire lake. There was about 400 individuals per square meter. So removing that truck removed a lot of substrate for zebra mussels. In 2025, Kiana shifted gears. We were trying some different methods. The lake was really green in 2025, so we couldn't visually scan as well as we could in 24. But again, the takeaway was places where there's human structures and places where we have rocks and gravel, we're tending to find a lot of zebra mussels. Kiana is continuing on with us again this year, and we're hoping to get our villager traps or our baby zebra mussels before they attach to a surface. They live in the water column for a while, so we're hoping to get our traps out earlier so she can look at zebra mussels. And then last, I wanted to end with a kind of a more positive story. So 2 summers ago, Isabel came to Stout from Virginia Tech as part of the Lakes REU, and she was really excited about plant diversity. So she knew that there was an invasive species, curly-leaf pondweed, that's found in Lake Menomin, and she wanted to do a survey of diversity of those different plants in the lake. These aquatic plants, oftentimes they get a rap as being a nuisance, right? When you're swimming, it's kind of gross to have weeds tangling around your legs. They can get caught in your boat motor. Fishing lines, fishing hooks can get trapped, but they are a critical part of a lake ecosystem. They pull nutrients away from algae, so lakes with a lot of aquatic plants often have less algae, less of those harmful algal blooms. They stabilize sediment to prevent nutrient release, and they're important for fish habitat as well. And so she went out and she collected plants from a whole bunch of different places around Lake Menomin, and these are her results. So each pie wedge represents a single species. Wider pie wedge equals more abundance. And so her big takeaways were we have this pretty diverse community of aquatic plants that are living in Lake Menomin. And even though we have this invasive species, Potamogeton crispus, or curly-leaf pondweed, it's only a really small part of the total diversity of different plants that we have in the lake. And so even though the lake certainly has its challenges, there's also some kind of raises some shine about interesting community dynamics that are happening in it. So where do we go from here? Freshwater Collaborative of Wisconsin recently funded us for 6 more years, so we're hoping to continue having student interns do kind of exciting and novel research, as well as do develop important skills. We are going to continue doing a lot of what we've always done, so monitoring streams, monitoring lakes, monitoring those black ash swamps. but we're also involving more faculty members who are kind of expanding the range of different projects that we can do, right? So in western states, we know that beavers are actually really important for supporting trout populations. So we're gonna start looking at beavers' hydrology and impacts on trout in some of our local streams. We're also gonna have a faculty member who uses genetic tools join us to look at population dynamics of some of the different species, those beavers and those cyanobacteria. And the Red Cedar River Basin Monitoring Group started as a community effort. And so if you are monitoring or managing, please reach out. That's Keith's email address on there. We'd like to hear about what you are doing. And with that, we'll take questions.
Yeah, so we have plenty of time for questions, or a couple of minutes for questions. We have a few minutes for questions, and folks are floating around with mics, so just throw up a hand.
So I fish Lake Menomin. I hang out there. It's beautiful in the winter. It's beautiful in the spring, and it's lovely in the fall. People ask me, how can you eat fish out of that polluted lake? Would you consider this a term polluted lake? And is there any type of fish advisories for consumption compared to what you're finding with these aquatic plants and vegetation green? Does it affect the fish? What's in the fish?
Yeah, so I will say I am not aware of any of the fish eating restrictions on the lake, so there's definitely someone who's more informed about that than me. In regards to the cyanotoxins in particular, I do know that there is a little research that shows that if fish are living in a lake that has high concentrations of microcystin, there's potential for buildup in some of the fattier tissue, so brain and liver, but most of the time we don't eat brain and liver of fish. So as far as the research on that goes, at least with microcystin, it does not lead to kind of buildup in fish or concern about consuming fish muscle. Other toxins I'm not as familiar with.
I'm wondering if the cyanotoxin levels in the lakes where you guys do the monitoring reach a level that exceeds the EPA standard for having health effects. If the community has any way of letting folks know that they might not want to boat or swim on the lake until those levels have declined.
Yeah, and I think that is a really important part of this. Like, how do we communicate risk about interacting with those waters? So right now, there's the blue-green algae signs that show up on Menomin and Tainter, at least. But should there be more efforts to say, hey, maybe in August, this is not a great lake to be swimming in? Oftentimes, I don't know, have you seen the, I think it's the DNR has the, how can you check if it's a harmful algal bloom, where you just take a scoop of water, you shake it up, and if it floats to the surface, you probably shouldn't swim in it. So there are a lot of different communication tools out there to talk about harmful algal blooms, but I think that is important and something that I am not as versed in. But how do we start communicating this, that July might be good, August might not be? It's a great point.
Hey, so I noticed on the one graph you had, there was the blue and the pink and the orange. I noticed that the blue was in a specific area, even though it moved with the season. It was kind of on the north end of the lake. Also, I was wondering why. And I was also wondering with the mussels having an effect, if they might have something to do with it, and also the plant diversity, if there's certain plants. I'm just really curious on why that is the way it is, and then why the red areas, the areas that get bad, if we know why that. And then I also had another question with the skin and inhalation, which was, is there no recommendations because the science isn't in yet? Or is it safe?
All right, so I'm going to say not a toxicologist, and so I know there have been studies looking at inhalation and dermal contact, and some people get rashes when they touch it, but the vast majority of the papers that I have read, which again, there are limits to that, it's ingesting it is the issue at this point. And so I just have heard people talk about being concerned about inhalation, but there is not data to suggest that that is problematic yet. All right, I'm gonna also kind of start to walk through your questions, so let me know when I miss one. So in regards to the spatial patterns that we're seeing here, I don't know. I think that one of the reasons that it's often less toxic up here is because you have a lot of moving water, because that's where the red cedar flows into Menomin. Cyanobacteria tend to do better when water columns are stable. And so my guess is you just have this new water that's flushing in here. And then by the time the water spent a few days making its way through the river, there's more time for the blooms to build up in there. So that's my hypothesis about that. No, it's because microcystis and aphanizomenon, they make little gas bubbles inside of their cells that lets them float up to the top. And so they can float better when the water is still. As soon as you start mixing it, they kind of get mixed down into the water column, and they don't grow as fast then. My other theory about why we see those spatial patterns is wind. I had a student try to measure this, and the results were very inconclusive. So this is just me going against what the data said, but I really just think it's that the dominant wind direction, is eastward across the lake, and so you end up with a lot of these surface scums that kind of just move around with the wind. I would like to keep studying that because I think it's a wind thing, but yeah. I know you had other questions as well, but okay.
Couple of questions. Wondering how far up toward the headwaters you'll be doing some of the work on the black ash swamps and then the beaver impacts. And another thing, especially if you're going to be looking at those aspects, there may be changes in forest management practices because of potential removing the slash in the harvesting product for biofuels. Is there an opportunity for you to kind of measure the impact of what happens with changes in forestry?
So on the black ash swamps, the most I can comment on is we will probably stick around where we have been working, which is mostly Muddy Creek and out at Gilbert Creek in the Triangle. And part of that is I would like to be doing broader watershed stuff. Our funding for this is primarily to paying the students, and so we get sort of limited in like how far can you go, and so it would be nice to do a lot broader coverage. But I will pass along to Mandy some of since she's leading that project, that she might look into places where there's forest management activities planned as a potential option.
The beaver question. Yeah, so we are pretty restricted. So this is Dr. Beston. She's pretty restricted to where there are dams, where people are willing to let her go out and look at the dams and sample those. So at some point, there will probably be a call on social media looking for people that have beaver dams, know where beaver dams or lodges are, and request to let us come and sample those because that would be great.
We have a lake home in Minnesota, and the zebra mussels arrived 10 plus years ago. And anecdotally, and observationally, just off my dock, the water clarity has improved because of the zebra mussels. What impact do you expect here now that the zebra mussels have arrived?
All right, so my guess is we don't have a ton of habitat for them here, right? We have a lot of mucky bottom. So will there be an impact on water clarity? Potentially, but I don't know if it'll be as big as in scope as it is in a lot of lakes where you have just a lot more rocky substrate for them. The other thing that I want to talk about is there is also a relationship where more zebra mussels leads to more toxicity of cyanobacteria, because you essentially filter out everybody that's not floating at the surface, and so the folks that are floating floating at the surface, those microcystis and aphanizomenon, those are our toxin producers. But then also they concentrate nutrients down at the bottom of the lake as well. And unfortunately, a lot of the different species of kind of cyanobacteria that grow down at the bottom of the lake can potentially also be toxin producers. So there's some interesting research that came out of Michigan when they first started seeing zebra mussels there a couple decades ago that we saw these pretty significant community shifts. Maybe that's changed in the last few decades, but yeah, CAN is actually interested in looking at that. So we're going to be looking at if in areas where there's high zebra mussels, if we see kind of less non-cyanobacteria. So, question?
All right, if you could all join me in thanking Dr. Gilland and Dr. Hayes one more time.